for those of you not familiar with MDIC. So it's the Medical Device Innovation Consortium, and we are a not-for-profit public-private partnership created with the main objective of advancing regulatory science. And I say the main objective, it used to be sole objective, but we're really starting to branch out and looking at the application of some of the work that we're doing beyond the regulatory sphere and into other parts of the healthcare ecosystem. But really focused on the advancing the regulatory science of medical devices for optimal patient benefit. And we do this through a number of projects that are ongoing at the organization. I'll get into that in the next slide. But overall, we are, we're a membership-based organization. We have about 76 members right now, and that includes the government, industry, and not-for-profits. The way that we work is we're a convener, and we bring in subject matter experts for whatever we're working on. And right now we have involved in all the projects that we're doing 900 plus subject matter experts. And then the output of our projects are varied. This project is going to be samples, but in a lot of cases we come out with frameworks or documents that go along with guidances or regulatory policy. You can look at our website and you can look at the kind of resources, the documents and the frameworks that we have developed over the past. And right now we have work groups that start and they sunset, but right now we have 61 active work groups, the one of which is the Somatic Reference Sample Initiative. And just to give you an idea of kind of the program areas, so the Somatic Reference Sample Initiative is under clinical diagnostics, and we have a number of programs in AI/ML. We had a COVID project and actually starting a number of new projects that I can talk about in a next incarnation. But we have, in addition to clinical diagnostics, we have clinical science, as well as digital health and technology, which, as you can imagine, is extremely active right now. And then we also have a NIST coordinating center that is focused on the use of real-world evidence for clinical applications. So that's kind of an overview of what our program areas and how we operate. And now we can focus on the Somatic Reference Sample Initiative that is part of the clinical diagnostics program. Of course, it's not moving ahead here. Hold on. Okay, so in presenting this project, I wanted to give a little bit of context in terms of how this project originated, or the idea for this project originated. So we have to go back to in 2018 when NGS was actually coming up as a very powerful technology for healthcare applications. And the NIH and FDA got together in a joint committee, and they're looking at, okay, what is necessary in order to support this as a technology for diagnostics. And so there were, three main areas, and none of these are going to be surprises, but it does set the stage for the initiation of this project. Is that they came up with that we need physical reference samples, tumor normal pairs. We need reference samples that are representative of the disease and the variant types. Reference samples that not only reflected frequent diseases that were high prevalence, but also rarer diseases, and then also in silico reference material. So not only the actual physical sample, but the bioinformatics and the data that's generated from the samples themselves. And of course, the tools are necessary for data analysis and interpretation, data integration, and then data distribution and analysis models. So all the tools to analyze the data and do the bioinformatics analysis. And then, of course, the other third leg to the stool is infrastructure. So the ability to have and the availability of high capacity platforms for storage and analysis of different types of data. And then, to make sure that there's appropriate policies and control in place to, ensure responsible data sharing. So with that,
pocket and present what we've learned so far and get your input about it.
Okay, so I'm going to go on to the next slide. So at the kind of in the same time frame, we had the FDA came out with this vision for regulation of NGS-based in vitro diagnostics, and this is taken from the FDA website on precision medicine. And so they were looking at, again, what from the regulatory side, what was needed to help support the development of and validation of NGS technology. And so one was the technical and analytical standards for next generation sequencing. And these are, if you think about like consensus standards that could be developed by the scientific community and then updated as science and technology advance and provide a path to market. Again, the use of FDA recognized databases to provide the clinical evidence. So kind of continuing on that theme of databases that were available for developers to use. And then in particular, as it pertains to the SRS project, develop, characterize, and make publicly available reference samples that can be used to support validation. And so, you'd have the samples and the sequence, the data sets, and then you would have a truth sequence that was available. So you knew what was, kind of using the sample, you knew what the sequence was, and that needed to be generated in order to be considered accurate. So then, with this, kind of stage set for needs and gaps and considerations. The Somatic Reference Samples Initiative, the idea for this was initiated. And the goal of this was to develop reference samples that can be available to the public. And again, for validation, but to improve the accuracy, reliability, and transparency of NGS-based oncology tests. And there was a decision early on that the focus of this was going to be targeted to oncology. And so, the path for this and the model for this is, what MDIC does is we're the convener and pulling in together all the stakeholders, the parts of the ecosystem that touch on and are affected by the diagnostics and NGS-based diagnostics. So pulling in the FDA, medical centers and laboratories that are doing the test, pharma that benefit from this test if they have a companion diagnostic, HHS and the payers that could benefit from this type of validation. Of course, the medical device industry as the developers of the technology, and then the organizations that are integral to medical devices. And really kind of working with that spectrum of stakeholders and really driving towards an impact to make test development and validation more efficient, streamline and possibly obviate steps in regulatory process for the diagnostics developers. And of course, to provide transparency, if we have these, defined samples, kind of developers and sponsors know what they need to use and what these are in order to test for accuracy. And then hopefully compress development times for the targeted therapeutics development. If you can get the diagnostic tests developed faster and more efficiently, then that has a downstream effect for the therapeutics that could be associated with that. So in the early stages of this project development, there was I think this was a kind of a milestone that really launched the project as it's known today. And we had, or there was in December, or sorry, mid 2018, there was a meeting at FDA to start scoping out this project. And over 30 organizations, again, all the stakeholders in the different groups in this ecosystem were represented, and came together and started scoping out, okay, how would we go about this, and how would we work together to make this happen, that we could develop these reference samples. And so I'm not going to go through you can see all the logos of the companies that were involved and everything, but this is me right here. I mentioned that.
Okay.
I mentioned that.
The text message yet?
Somebody's not
yet.
on mute.
Not yet.
All right, so so this is me. So when I had the opportunity and the privilege of actually being invited to this organizational meeting and participating as an industry representative. As you know, ATCC is very involved with and interested in the standards, and so I was able to participate in this, and then kind of lo and behold, a few years later, I'm actually back and really excited to be leading the project. So what was decided is, how are we going to do this? Is that the approach was, and it's based on the model of how MDIC does things, is engaging the scientific community, collaboratively developing and characterizing, and of course, making public these reference samples together with data sets that would support the validation and the potential to improve the regulatory approval process of NGS-based diagnostic tests. So with the initial cancer focus being, or identified that, cancer was going to be the initial focus, the decision is that we were going to use the highly characterized cell lines from Genome in a Bottle. And I heard from Mickey right before I started today that you guys have heard from Justin Zook, who was leading the Genome in a Bottle initiative at NIST, where they have sequenced the genomes from an Ashkenazi Jewish family. They're very highly characterized cell lines. And so using that as the background, the decision was made to use that and then introduce cancer variants, define cancer variants into these cell lines by CRISPR gene editing, so that you have a very deliberate and defined sequence change that you're putting into these cell lines, so that you that represent cancer variants and that can be the basis of these reference samples. And so in the early phases of this, they were looking for funding and were able to get the funding from IVD manufacturers and also some philanthropic organizations that recognize the patient benefit of this effort. We got a fully funded pilot for creating 10 variants. Initial scope was a little bit like 5 times as large as that, but I think I think this is the better way to go. That it was kind of carved down to a pilot, and that we could get a lot of lessons learned from this. And then once we complete this pilot, then we can expand on to future, including future variants and expanding the scope of the project. So the pilot objectives were engineer 10 cell lines using CRISPR. And each containing one gene variant from a prioritized list of variants. And I'll go into how we prioritized or how we selected those variants in some slides that are coming up. And another aspect of this, which I think is really important, and I know at least, I know some of the work that Mickey works on, and I know there's a lot of reference material or reference sample interest or use, is that using this project to look at iterative batches, because very rarely can you develop a reference material that is an infinite source, and you have to come up with a strategy where you continue to make batches to meet the continuing demand over time for reference samples. So we're using this project as an opportunity to look at batch-to-batch variability and how to control for that, and how to ensure that the reference material is standardized and the utility is maintained and the integrity is maintained from batch to batch. So the idea is that we produce these cell lines, and then we do mixtures, put them together in mixtures for the final product in FFPE. And I don't know, how many of you are aware of this particular format, but it's formalin fixed, paraffin embedded format. And this is often what is used for if tissue samples, if they're taking a biopsy, that they would take tissue from a patient, they would embed this in this format in formalin fixed, paraffin embedded. And then they would take a slice of that and do the sequencing or the analysis in order to do the diagnosis. So what we're doing is the plan is that we're mixing these cell lines together. Those mixtures will be embedded in the formalin. And then those slices would be used as these contrived samples that would mimic human samples and would be used for the validation to ensure that the accuracy of the sequence read in order to ensure that you get good accurate diagnoses. So then also in this project is, the objectives are to develop efficiencies in validation processes for reference samples. We see this as a model that can potentially be used for the development of additional reference samples, as I've mentioned. So using this as a way to develop efficiencies and lessons learned for validation of these reference samples. And then establish a publicly available and authoritatively characterized data sets. And this is really an important adjunct to the samples themselves, is to have the bioinformatic analyses done and the data sets available to use in conjunction with the samples for validation. And of course, identify future targets. Take these lessons learned and then identify the next future targets, whether it be in cancer or infectious disease or even many different pathways for that. So I wanted to get to the variant selection because this was important. Since we're doing 10, we wanted to make sure that we were selecting variants that were highly correlated with disease. And so this is a evidence pyramid that is taken from the FDA website again, and looking at the levels of evidence, and you have 3 levels that have been identified. One is a level where companion diagnostics have been developed against that variant to detect that variant, so very high level, and there's most likely a drug that's associated, a companion diagnostic drug that's associated with that. Then level 2 is cancer mutations that have evidence of clinical significance. So there is enough significance to associate that variant with disease. And then level 3 is mutations that have potential significance. So what we wanted to do is make sure that this work was focused on ones that had a certain level of significance. And so we decided that we would only look at and evaluate variants that fit into the level 1 and level 2 categories. And then, so to continue on the variant selection, we had, as part of the first phase of this project, the SRS project, is that we had a landscape analysis report that was published on our website in 2019, and that was kind of a landscape analysis of the variants that have been identified. It's by now it's out of date because there's always new variants that are associated with disease, but that was up to date at the time, and then also any reference materials that had already been developed and that were valuable, because one of the things is we wanted to make sure that we weren't duplicating efforts. So from that landscape report, and also with input from the FDA, and we were working closely with a sub-team of the steering committee, is that we were, initially when we were based on levels 1 and 2 of evidence levels 1 and 2, we had over 600 variants. We sent it out for public comment and did some pairing of it. We took out anything that was associated with a blood cancer, just to kind of limit the field for this project. We came up with a final list of 54 variants, and then from that 54, we needed to select 10. So enter our SRS task force on variant prioritization, and looking at how to hone in on 10 from the list of 54. This task force really focused in on the following criteria. One was a higher level of correlation with disease, a higher correlation of clinical significance, and this is OncoKB level 1, which is a level identified in the OncoKB database. And this is genes for which specific alterations have been recognized by the FDA as predictive of response to an FDA-approved drug. So slightly higher clinical evidence criteria. But in addition to that, there were also other considerations and other criteria. We wanted to make sure that we captured a range of mutation types in the variants that we were going to engineer into these cell lines. And so we wanted to, as best as possible, to try to include insertions, deletions, any structural rearrangements, nucleotide variants, and again, because we wanted to maximize lessons learned, and so let's try all these different types and see what we learn from that, or if some are better, or how to maybe overcome any overcome some technical difficulty. Speaking of technical difficulties, we wanted to get a range of technical difficulties, just in terms of, for again, technical learning in the manufacturing. The technical difficulties do correlate somewhat with mutation types, in that, for example, structural rearrangements are generally harder to gene edit into a cell line than like a single nucleotide variant. But we wanted to make sure that those were represented in the 10 variants that were part of the pilot project. And also, likewise, we wanted to get a range of variant prevalence and a range of cancer types. So we felt like that the set that we were doing kind of represent as broad as possible the possibilities for variants, cancer variants, and where we can maximize the lessons learned on a number of fronts. So what we came up with is actually two groups of 10. There's a top 10 group and then a backup 10. And the reason for this is I don't know how many people are familiar with the CRISPR technology, but there's a lot of different steps. There's some technical challenges, and you can't always predict whether a particular or specific mutation that you're trying to introduce will work or not. So we had to have a backup list. And the idea is that we would, if we tried, we would, start with, a set of 10, and then if we ran into a problem that let's say with FGFR3, then we would go to the backup on a similar variant type, another structural rearrangement, and then try our best and start engineering, doing CRISPR and trying to do that one. So with the backup list, we had something to go to immediately that everybody agreed to, and we wouldn't have to do an assessment whenever we ran into a technical difficulty. So as you can see, they represent different variance types. What you also might notice is that there's no copy number variants in here. And that is a result of the CRISPR it's been notoriously difficult to do copy number variants with CRISPR. So we might address that at another time in another phase with a different technology, but to be determined. But right now we feel, with structural rearrangements, we have insertion deletions represented, and several different types of nucleotide variants represented as well in both top 10 and the backup. And again, representing a number of different cancers. So I'll put this all in. So the variants, the variants were identified, and they were prioritized, and so we have our list. And so now we were able to give that information to our manufacturing partner, which is Horizon Discovery. Well, they're now part of PerkinElmer. But the manufacturing approach is that, and I've mentioned that we're using the genome in a bottle parental cell line, that we will take that, or I say the royal we, the Horizon's doing this, is that we would engineer the variant 1 into one cell line, 2 into a second cell line, and so on, so that the result is that we are going to get 10 cell lines, each with a single variant. Then the next step is that those cell lines are mixed in, maybe 2 or 3. I have 3 represented here. That mixing is going to depend on a lot on allele frequency and other considerations. but they will be mixed so that you have representation of 2 to 3 or 3 to 5 different variants in a cell line mixture. They will then be embedded in formalin, and the FFPE blocks will be generated. Then the FFPE sections will be derived, and those will be what will be available for commercial distribution. So again, those sections are mimicking the human tissue section that would be used for diagnosis. Then another piece of this is that, again, I keep come back to this, is that there are a second batch will be done, and then significant analysis will be performed to look at the first batch versus the second batch and how to control for any variation. We are generating the genomic DNA and RNA from these different parts of the process, and as well as the FFP sections. And then that material is going to, MDIC has bioinformaticians working closely with NIST to do the data validation, data integration. And we have a whole plan that involves other collaborators, other sequence platform and laboratories, other collaborators to support that effort to do the validation and integration. So what they're doing right now is, while they're waiting for the samples to be generated, to be manufactured, which is in process, they're working on the parental cell lines to develop benchmarks and analyses approaches for the parental cell line HG002, which is a genome in the bottle cell line. And they're doing a lot of analysis, background analysis, so that that's completed. And then when we get the engineered variants into the cell lines, that we completely understand the background and we know what we're going to be doing for the analyses approaches for the bioinformatics for the reference samples. And then when they do get the samples, then the primary goal is to experimentally verify the locations of the mutations using multiple technologies and orthogonal methods. And as important as that is, because we need to make sure the variants are engineered correctly and in the right location, we have to evaluate off-target effects. And again, in the CRISPR technology, the good part about it and the strength of this is that you can do very targeted mutations, but there also are potentially off-target effects that need to be evaluated to ensure that they're not affecting the use of this material as a reference material. If it was, if there were mutations or changes that were introduced that affected its ability to grow in culture or something to that effect, where it reduces the effectiveness of that sample or that that cell line, then we'd have to take another approach. So it's really important to evaluate the off-target effects as well. And then we need to confirm the performance in multiple NGS diagnostic platforms. We want to have it as broad as kind of a test study as possible, looking at the performance in the different platforms. And then also develop the data sets, the complete and validated and characterized data sets for ongoing research and regulatory applications. And then again, perform extensive cross-batch analysis to define protocol for ongoing validation efforts and efficiencies. So the timeline for this is, again, I mentioned that the first meeting was June 2018 at the FDA, and that was considered the project launch. In March of 2019, we had that landscape analysis published. And then between the years of 2019 and 2021, we were working on the project plan and developing that project plan, getting funding secured for the pilot, and finding a manufacturing partner. And that was all completed by the end of October 2021. So then we were able to actually kick off phase 2 in January of 2022. In May, we were able to have contracts completed. And then also we also confirmed the priority variance, and that's when the data and analysis efforts were initiated. And I've been talking about the technical difficulties of CRISPR, but you don't know how excited we were that we finally got to, actually get over the administrative difficulties of getting all the contracts in place. And I'm sure some of you understand that. But that was a major milestone in May when we had the contracts in place and we were able to actually launch on the manufacturing. What we're expecting is that at the end of this year, that we are looking at the manufacturing being complete for these 10 samples. Knock on wood for that. There's a lot of, as I mentioned, this is a, kind of biological process. So with the cell lines, so, there might be some technical hurdles that might change that timeline. But if we keep to that, we are looking at September of 2024 commercial availability. So between the end of this year and when they're available, that's when NIST and MDIC would be completing the bioinformatics analysis and completing the data sets that accompany the samples. Okay, so what I've presented so far is the driver of this project was utility in regulatory evaluation. So the FDA had a gap in recognizing this as NGS is a huge a very powerful diagnostic tool, but how do we regulate that and how do we evaluate performance? And so this is a piece of the puzzle and a tool in the tool chest that can help potentially help support regulatory decision making. But I wanted to show the potential impact of these reference samples across the total diagnostic product life cycle. And I borrowed this figure from an article in the Journal of Medical Devices, where it is showing the product development life cycle, where you have the discovery and ideation of a medical device, or in this case IVD. Invention and prototyping, then you go to preclinical studies, to clinical studies. And again, this can be iterative as you fine tune your device. And then you go into regulatory decision making, which we've talked, this whole talk has been really on how to improve and use these tools to improve and expedite the approval process. And then you go into product launch and then to post-market efforts. And it struck me that there is utility, and it's very evident to the SRS steering committee that these tools really can have utility throughout the development process. And just wanted to highlight these a little bit in terms of discovery and ideation. When you're thinking about as a developer that you want to, is there an opportunity or is there a market need for this device I'm developing? You could use these samples to understand the market need and the competitive landscape. If reference samples in general are, have the true sequence, or in the case of NGS, a true sequence, then you could evaluate what's out there. Is the performance acceptable? Is there room for improvement? And is there a market need for improved technology? Then in early technical, in invention and prototyping, you can use these to do early technical validation, as well as use in early feasibility studies and preclinical. And when you get to the clinical, these samples can be used to streamline clinical trial eligibility. And one of the issues and challenges that we've heard about is, especially when it comes to inclusion or exclusion into a clinical trial, is that you have to go to that trial and those laboratories in order to get the sequencing done that they will accept, because there's still differences in the way that sequencing platforms are performing. and there's lack of kind of the true sequence. Right now, you have to, you may have to repeat your studies, and there is some unknown about the accuracy of that sequencing. With these samples, since the truth sequence is known, then this would really optimize and improve the understanding and help out with clinical trial eligibility. We've already talked about expediting the approval process, but then we're just starting to have the discussions with payers about, can these tools be used for as an objective tool in evaluating performance in order to make coverage decisions. And we've had some, early conversations that with payers that this is the way that they're going. They need a way, especially for laboratory developed tests, that they need a way, to not evaluate the lab itself, but evaluate the lab performance on a particular assay. And so really putting into place the use of these types of analytical tools in order to make decisions. And so we've had conversations about and looking forward to beta tests with these samples with payers in order to help evaluate NGS platforms for diagnostic applications. And then in post-market, that one of the advantages of these samples is that with the truth now, what the result should be with the reference samples, you can have higher confidence in the diagnosis and the sequencing of a patient sample. And so then that you're getting them, not only are they going into the right clinical trials, but then that they're being targeted for the right companion drug. And so you have much more accurate post-market information on efficacy for pharmaceuticals. You also can use these samples and the data sets from these for artificial intelligence and machine learning truth training. But most importantly, we talk about the diagnostic product lifecycle, but most importantly, we should be talking about the patient. And now the patient with if next generation sequencing platforms are sponsors and developers use these reference samples to develop and approve these platforms, then you're sure of your sequence. Then you not only can you have lab-to-lab portability, but now you thoroughly understand your therapy options with confidence in what your sequence, what the result of your sequence was. So all along, most importantly for the patient, but all along throughout the product life cycle, there's application for these reference samples. Maybe in slightly different formats, not FFPE, but there is a potential role throughout the life cycle of the utility of these samples. So that's kind of the overview of kind of where we are in the process, what we're doing, and the utility of these samples. But in summary, what we are looking at, what the effort is to deliver authoritatively characterized and validated reference samples for NGS-based diagnostics, along with the data sets. And one of the criteria that at the start is that they would have availability, that we would have initial reference samples, and then new batches would be delivered and manufactured as needed from common source material. Accessibility, and this is still in the work. This has to be it's definitely a tool, but it has to be an affordable tool to members of the community. There has to be recognition that if these are to be used everywhere, that they have to be at a level where they're available and accessible to the majority of the scientific community. They have the potential to transform the regulatory review process. There's still work to be done on that from an FDA perspective. And then, there's downstream effects to improve therapeutic development and approval. But one of the things that is under discussion right now is that, are these reference samples candidates for FDA's medical device tool development program? And I'm not going to go into too much detail on this, but this has been an interesting discussion within the steering committee and MDIC. We were not aware of this really as it applies to reference samples. But there is some interest from the FDA's perspective and potentially ours, if this is, kind of it's kind of a de facto approval, if that we get recognized as a medical device development tool, that could make the uptake of these more broad within the scientific and medical community. And if anybody wants to look at that, you can look. The FDA has a website on that. Typically, it hasn't been reference samples that have been recognized as such, but there is some interest from the FDA to see if potentially we can make this work. So there's ongoing work with MDIC, with the SRS team, and with FDA to see if this is a good fit. Then also, this is also a project that can be the basis of building a sustainable model for reference samples in general, or somatic reference samples and data set generation, and then to help determine the next set of SRS would be, a phase 3. And, obviously there's an expansion in cancer, but it also could apply to other diseases. So looking at other opportunities, even as this project is ongoing, as to other interested and applicable areas for expansion. And then just the emphasis that there is, and I showed that in the previous slide, utility beyond regulatory, and looking at, again, projects and interactions and partnerships that could help have these as tools to support development, reimbursement, and disease management. So I think that we are at the end of my talk, and really appreciate, again, thank you for the invitation. Enjoyed presenting to the group in Frederick, and happy to take any questions at this point.
Thank you very much, Mary Ellen. Can you hear me?
I can hear you loud and clear.
Okay, great. Maybe I'll jump right in and ask a question. I think CAP was at the table, the College of American Pathology. These seem like they would be a wonderful addition to their proficiency testing menu. Have they been updated recently, and are they interested?
Yeah, so it's very timely question, Mickey. So, as I mentioned, we've been having these discussions with payers. We just started the discussions with payers, and have heard this incredible interest in having these used as tools for reimbursement decisions. And so they're looking at, what we hear from their perspective is that they're looking for, additional that, CAP and the proficiency testing that is ongoing for CLIA certification for laboratories is important and, effective to a certain degree, but, kind of further work with including these types of reference samples as proficiency testing, like as blind test, would be extremely valuable for a number of, for the patient, for the payers, for a number of stakeholders. So that's the long answer. The short answer is that CAP, we have to reconnect with CAP now, and they're aware of it. I've been having unofficial and casual conversations, but it's time to kind of bring this up and talk about either integrating or being an adjunct to proficiency testing.
Excellent, thank you. Folks, the floor is open. Anybody have a question, please unmute yourself and jump right in.
Yeah, I have a, Mary Ellen, that was really, really impressive. Congratulations. I hope it goes the distance.
Me too. Yeah.
But it looks really promising. Here's kind of a naive question. I mean, obviously, genome in a bottle versus what you guys are doing, you know, you've kind of taken it to at least several levels beyond that. But what are the scenarios, if there are any, where you could use just the, you know, plain old genome in the bottle reference standard versus what you guys are developing, or could you even use it? How do the scenarios differ in terms of applicability?
Yeah, so I think you'd have to, I don't know, and Mickey maybe knows more about me. I don't know what the genome is of the Ashkenazi. I know that they have variants in them and everything, but this is our approach is, it's almost not paying attention to what's there. It's just using that as a background and deliberately putting in variants that have utility, and we can, you know, we know exactly where they are. We know we can measure them. We know what's around them. So I imagine that there would be limited utility. There could be some utility if you just wanted variants that were in their, you know, in their germline DNA. But you know, we haven't really talked about that, and I think it's that would be more kind of a byproduct than a deliberate you know, approach. And Mickey, I don't know if you have more comments about that.
I would jump in and say, yes, Len, that the genome in the bottle is a normal genome, obviously.
Yeah.
So it's relevant variants for oncology. Yeah. It's as the most wonderful specificity material. If we're looking, does an assay hold false positives? These genomes are so well characterized that if you see variants that you aren't expecting, you know, you've got a weak link, a bad, difficult to sequence region, which there are many in the genome, obviously. So they serve very good there. And by knocking in now oncology relevant variants, it gives us, at least in oncology, the ideal scenario. The other thing is, by doing the mixtures, we can take down the allele frequencies that would be representative of what we see in tumor specimens, where you have stroma coming along for the ride, and sometimes clonal variants that you're making claims that your assay can detect 5%. Well, we'll have truth at 5% and have at it.
Great, thank you. Anyway, it looks terrifically promising.
I'm excited. I'm really excited about this. This is and it seems like that, you know, and all the partners are. Mickey was on the call with the payers, and to hear them going in this direction too, where there's a lot of analytical validation that's going to be required, is exciting. Any other questions?
DJ, do you want to know when you can get your hands on these, even in a pre-code? I'll answer the question for him. Yes, we would love to get our hands. I've been talking to Mary Ellen about Combo Match, which is a national study that we've been engaged in. And we have 40 independent lab developed tests. And heaven forbid, worrying about how concordant these different assays are. These materials would be a godsend for doing the things like Mary Ellen was talking about, for looking across the assays and their performance.
Yeah. Hey, Mickey's got an inside line, so.
Yeah. Maggie Scully has a question in the chat. Want me to read it?
Hold on.
Sorry, I'm here. I thank you so much for your talk. That was really informative. Discussed sort of that extensive review needed to select the, you know, clinically relevant variants, and the landscape review that you performed, and the tiering. I want to know if there is an up-to-date source of that, and is there someone that maintains that?
Yeah, there is, and I can put that in the chat, or when we're done. I have to find it, but yes, there is.
Appreciate it. Thank you.
Yep. Great question. It looks like,
Yeah, DJ is waving his hand at us, but I don't think he can get off mute, and I don't know how to do that for him.
yeah, I don't know how to do that either. Let me see.
DJ, you could put any comments or questions in chat if that works. Okay, something's on the way, it looks like. So maybe to give you a tough question while we're waiting for DJ.
Yeah.
B cells certainly aren't lung cancer tissue. And in some of our interactions with the FDA, we get challenged with the matrix. And are you using materials for your validation that are from the clinical matrix? Have they mentioned anything to you about this? Because it depends on who you talk to. Somebody will think of it one day and challenge you. And I'm just curious, has that come up yet?
You know, that hasn't come up yet, but you know, I think there's definitely more work that right now we're just focused on they're doing the analysis on the you know, the parental cell lines. We have a lot more work to do to kind of scope out and you know, come up with the approach for validation of you know, patient samples. And so I think that's a great point that you made, Mickey, and I think we have to consider that when we're doing this.
Yeah. Okay.
Thanks.
And it doesn't look like DJ is getting through, and we're getting close to the
11:59.
Any final
Oh, there we
questions?
go.
Well, I do know how to get a hold of DJ.
So we just got one. It says, do you plan to target allele frequencies at a range that is compatible with MR? Yes. That is definitely, you know, that we want to make sure that we're able to, you know, I think we're going to have some allele frequencies that have a certain basement level, but that are at the levels of, you know, that are usual with diagnostic testing.
Yep. Okay. Very good. Well, I want to just finish up by once again thanking you. It's a wonderful program. We look forward to the commercialization and keeping in touch. So I thank you for your time and educating us on this topic.
Thanks so much for inviting me. I had a great time presenting, and thanks for your input and your involvement, Mickey.
Pleasures. Thank
Okay.
you.
Thank you, Mary Ellen.
Okay, great seeing you. Great seeing you, Len. I'll follow up with you. We got to get together.
Great.
Okay. Take care, everyone. Thanks so much.
Thanks.
Yep, thank you.
Goodbye.